Test. Jamie, can you hear this? Yes. Testing.
Yes, I can.
Audio.
Yes, I can hear you. I can hear you fine.
You hearing anything?
Yes, I can hear you fine, Brad. Okay.
Putting one in the mic. How do you like to look at the screen or like? Yeah. Okay.
Welcome, everyone. I hope you can hear me. Can you hear me? Thumbs up, thumbs down? Okay. We're going to get started in just a few minutes, so gather together whatever you want to eat and snack on. And we will have Sara coming up in a second. We'll introduce her, but just a few minutes. Get yourself settled.
Okay.
All right, welcome everyone. We're going to start the presentation pretty soon. I'm Brad Smith, our president couldn't be here today, so we're going to do the best we can without Jamie, and he's watching with us on Zoom. So we're going to change it up a little this year. We're going to go right to the keynote right away instead of making you wait for 2 or 3 hours. So we're going to have Sara give us a nice chestnut presentation here on what it's really going to take, I think, to bring back chestnut to the forest. And I've seen Sara in the organization, what, 25 years or something? Yeah, at least 20 years. So she's been working for a long time. I have a nice little biography of her, which is in extremely small print. So I'll read it as best I can. Sara Fitzsimmons has worked with the American Chestnut Foundation at Penn State University since 2003, assisting chestnut growers and researchers throughout the Appalachian Mountains. Born and raised in southern West Virginia, Hinton actually, Sara studied biology at Drew University in Madison, New Jersey. She then received a master's degree in forest ecology and resource management from Duke University, St. Nicholas School. After a short stint as an editorial assistant at Beer Magazine, Sara returned to the forestry field, where she has been ever since. Sara hopes her research and professional work will facilitate long-term conservation and restoration of native tree species at risk from exotic pests and then my thing blank got cut off, so whatever, bad things. Here is Sara.
Hi, everybody. Can I take this off? Yeah. Thanks for having me up in New England. So I went and did the main chapter meeting a couple of weeks ago, which was fantastic. I had an opportunity to run around Maine, and I'm going to show you guys a couple pictures from that trip. I did not have a chance to visit any of the Massachusetts chapter orchards on this trip. My family's actually been with me on this trip. So we went to Dinosaur State Park in Connecticut yesterday, which has nothing to do with chestnuts, but was a ton of fun. Again, thank you guys for giving me the opportunity to come up here and spend some time with you and talk a little bit about restoration of American chestnut. As Brad said, I've worked with the foundation for about 20 years. I started as an intern when I was doing my master's work at Duke, and I've been here ever since. And my title right now is Director of Restoration. So that's really what I do on a day-to-day basis. I help assist citizen scientists like yourself, people who work with nonprofit organizations. I work with different researchers to try and figure out what are the different kinds of studies we need to put chestnut back in the forest. How much is it going to take? What about the population genetics? A lot of time, the foundation has spent the past 35, almost 40 years trying to figure out how to get resistance into the tree. And our next job is really how do we get this tree back out in the woods? And so a lot of the work that we've done so far has been silvicultural in nature. We've worked with a lot of foresters to do silvicultural trials where we take material that's not quite resistant or we don't know if it's resistant enough, but we've learned a lot from this material. And so a lot of what I'm going to show today is what we've learned about so far. There's still lots of questions out there, but the good thing is we have time, for better or worse, to work this out. Yeah, foresters kind of think on a different time scale. And I know I've gained a lot of patience, which I think my kids might appreciate, working in forest breeding. Forestry breeding definitely requires having some patience and working on a bit of a different time scale than maybe the current society, I guess, would prefer. So the first, how do we make a disease-resistant American chestnut? And so this was, I think, one of Kendra's first pictures. This is the Kendra pose up in the upper right-hand corner. So if you ever have a chance to visit a large American chestnut tree like this one or any large tree, that's the pose that you are required to do. It's leg up, arms around, that's the Kendra pose. So this was a tree in Berlin, Vermont, one of the largest American chestnuts. Unfortunately, it's gotten the blight and died now. But that's still a really inspirational size tree. The Chestnut Foundation worked for a long time focusing on backcross breeding, and that's what the Massachusetts Rhode Island chapter has done a lot of work toward. And more recently, the foundation has adopted a methodology called 3-Bur. And so 3-Bur is basically the integration of all the different technologies that we can use to bring disease resistance into a self-sustaining American chestnut population. So the 3 Bs are breeding, biotechnology, and biocontrol. And I love this acronym of 3-Bur because there are 3 chestnuts in a chestnut burr, and there are 3 Bs in our strategy. We've got the traditional breeding like backcross breeding and breeding together American chestnuts for their resistance. The biotech, you all probably are familiar with the Darling 58 and the genetic engineering that's been done for that work. There are other methods of biotechnology like gene editing. CRISPR is a big hot topic these days. That all falls under the biotech realm. And then biocontrol is where we try to make the fungus sick with some other biological organism. People have looked at other fungi, people have looked at bacteria. We're of course looking very closely at a virus. It's called hypovirulence, where the fungus gets infected by a virus to make it sick. All of these are different strategies that we can implement to try and restore the American chestnut. I love this picture too. So I started back in the year 2000. That was when I had my internship. And the people who mentored me are Ann and Bob Leffel, the one flanking Adam Carl there in the middle. Adam was an intern when I was an intern with the Pennsylvania, New Jersey chapter. Ann passed away last year, unfortunately. But I always like to show this picture because volunteers, citizen scientists are integral to making this work happen, not just the breeding work or the conservation work that we've done to date, but are going to be absolutely vital. We cannot do this without people and hands planting trees. So the chapter involvement is going to be absolutely critical for the next 40 plus years. And so we've been rolling out these 6 things that we'd really like the chapters to work on in the next few years. So 1, establish GCOs and maintain local diversity. And that's something that I know the Massachusetts Rhode Island chapter has really been doing a lot of recently. So a GCO is a germplasm conservation orchard. And basically a GCO is taking local American chestnuts, wild type American chestnuts, trying to get as diverse a population as possible from around the state or region, and then planting them in a single location. And the reason we do that is because it's a lot easier to use chestnuts in someone's backyard than it is to go from here to there, to there, to there, breeding on those trees. So that's why we want to take, usually a GCO is about 10 different genotypes. So taking 10 seeds from 10 different trees for about 100 trees, that's our typical GCO unit. It can be less, it can be more, but we like to go on about 100. That's a little less than an acre and is an easy unit to work with. The second thing that we want folks to work on is looking for best of the best hybrids and establishing production and breeding orchards of those. And again, I know the Massachusetts Rhode Island chapter is doing a lot of that work. So the Backross breeding program that the Chestnut Foundation spent a lot of time on, the original hope was that we would have hundreds of individuals that were highly blight resistant and have great American character. What we have found since then is that chestnut blight resistance is a lot more complicated than was once hypothesized. And so instead of hundreds of trees that have these, traits that we're looking for of high blight resistance and great American character, instead of hundreds, we're looking more at dozens. So we have a lot more selection and roguing work that we have to do. And a lot of that is being done right now through genotyping and through phenotyping hundreds and hundreds, thousands and thousands of trees. So that we've got our work ahead for that. So we want to find those trees, breed them together, and plant them in production orchards. The third thing is oxalic oxidase seed production. This is the transgenic tree that would be based first on Darling 58, and then after that on other types of transgenic lines that come out. So we need to establish seed orchards and then bank them for distribution upon deregulation. And I'm going to cover all of these in a little bit more detail in a minute. The fourth is reintroduction and restoration projects. So as I mentioned, the first step is to get disease-resistant material. The next step is how do you get these in the woods, which is no small feat. A lot of people think you can just plant a tree and walk away, and that's not the case. Certain types of lands and different types of cases require that you establish trees and maintain them in different ways. So while we have best management practices established, these may or may not be suitable for all types of locations all the time. So those top four are all sort of the scientific planting trees element. The last two are more of the outreach. How do we ensure that we've got people working on this program into the future? So number 5 is outreach to legislators. So that's something that the Chestnut Foundation has never really done. We have not reached out to local politicians. So every member, all of you live in a different congressional district, both at the state level and at the federal level. We need to have our local politicians know more about this work, not just chestnut, but putting this into the greater realm of forest health as a whole. American chestnut serves as a fantastic place to get a foot in the door to say, look, American chestnut was one of the first species that really was eradicated from our forests because of exotic invasive pathogens, but there are others out there that are under threat. Our beeches are under threat from beech bark disease. We've got our ashes being eradicated because of emerald ash borer. Hemlocks are being eradicated because of hemlock woolly adelgid. There are a lot of these pressures being put on our native forests. And so American chestnut can serve as the template of restoration, but we need folks to recognize that this is a greater forest health, greater forest sustainability issue than just American chestnut. So we really hope that our members will start talking more actively about these issues, and we can help give you talking points in that. If you can get a meeting, we would be more than happy to help coach you through that and give you talking points when you meet with them. And then number 6 is sort of the same thing. Tell the story. Tell the story of American chestnut to different audiences. I give talks all the time. I give them to Lions Club, to the Rotary Club, to school groups, to different hiking clubs, sportsmen's organizations. There's all kinds of different groups who are going to be interested in this story. And again, obviously we're talking about American chestnut. We're very focused on restoration of American chestnut, but I think to be more in broader terms, we're setting the stage for greater sustainability and greater forest health issues into the future. So let's go back here. So number 1, establishing GCOs and maintaining local diversity. The way that you do that is finding wild American chestnuts, which is not too hard in Massachusetts, believe it or not. How many of you guys have found local wild American chestnuts? So you guys know where they are. They're all over Massachusetts. And so the current status of American chestnut is that there are about 435 million American chestnuts still across the eastern United States. The problem is that, as you know, most of them are small sprouts. And so even though they are numerous, too numerous to have any sort of formal classification of endangered or extinct, we call them functionally extinct. And that's because they no longer serve the function in the ecosystem that they once did. So most of them are not producing nuts. If they do, it's for a very short period of time, and we take a lot of them. for our own purposes. But you can see here the areas where they used to be, where American chestnut used to be plentiful here in the southern Appalachians. So Massachusetts, Connecticut, southern New England, this has thousands, if not millions, of sprouts. Where I am in Pennsylvania, they're all over the place, especially along the Appalachian Trail. Western North Carolina, Eastern Tennessee, there are just hundreds of thousands, millions of trees in these locations. Where a majority of diversity for the species is the southern Appalachian Mountains. And this is where TACF is really focusing to try and find trees. The problem is they used to be there, and now there's very few of them for various reasons. But you can think of when the glaciers were at their last maximum, all the trees sought refuge down here. And so you typically find with most tree species, you find a lot of their diversity in the southern Appalachians. And unfortunately, that's the area that is most at risk due to climate change. So one of the things that we're hoping that New England regions might do is take trees, if we can find them, take grass, take nuts from these southern locations and plant them up here to conserve them long term, and also see how well adapted they are to these locations. So that's one thing that we're pushing right now, is not only finding local germplasm, but also trying to figure out how far can we actually move this material once we conserve it. And then just to give you a comparison, so this is based on forest inventory analysis data. This map over here is where we've had volunteers report locations of American chestnut. Whenever you see CADE, C-A-D-E, that's my shorthand for American chestnut. C-A is Castanea, D-E is dentata, so I just shorthand it CADE. But you can see, again, Massachusetts is really well represented. Connecticut, New Hampshire, we've got over 20 individuals across these areas. And then you can see it really peters out once you get down here into the southern Appalachians, and even into Indiana and Ohio, where they used to be plentiful. We are losing specimens. Large trees that our members used to know about in the 80s and 90s have disappeared. So we are kind of racing against the clock for conserving germplasm in the extremes of the range. Even though they're plentiful here, they aren't in other areas. This is one of the GCOs that I got to visit when I was in central Maine. This is up in Piscataquis County, near the town of Dover-Foxcroft. This is a GCO that was established. And again, this is a fairly simple, about 100 trees with an electric fence around it. And then a lot of people say, well, why would I plant American chestnuts? They're just going to get the blight and die. And that's true, but you can have really incredible trees for some amount of time. Most of the time, American chestnuts will grow for about 20 years, or you can get them to grow for about 20 years. These trees are about 22, 25 years old. This is Eric Evans. He's been involved with the Maine chapter for a long, long time. These are the trees that he grew from seeds from nearby in Maine. This is one of the trees that got the blight, and you can see how quickly it grew. And these are all the logs he got from a single tree. He's going to get them cut up and make some nice lumber that he's then going to sell, or make different types of wood products on. And you can see the blight, maybe you can't, but the blight is really proliferating amazingly here. But you can really get some impressive specimens from wild American chestnuts. And then of course they're going to resprout, and you can get chestnuts year after year, and we can also use them for breeding, diversifying blight-resistant materials. This is Tom Clack. He's a professor at the University of New England, and this is one of his GCOs in southern New England. And so number 3 on my list was diversifying and making seed orchards of OXO. So oxalic oxidase is the gene that's been put into Darling 58 to confer resistance. Well, what happens is when you take that tree, when you take that clone and you put it on American chestnut, only about 40% of them are actually resistant. So you have to do a lot of testing in order to figure out which ones have the genes and which ones don't. And that's what this process is here. You have to cut a little part of the nut off, put it into solution, and you can see here, this one is not resistant, this one is, and then you color code it, or at least Tom does, color codes it green for go. This one's resistant, we can plant it and know it's resistant. If it's not, then he paints it red, and we know that that's a non-transgenic counterpart. So, and this will come into play a little bit later when we talk about numbers. How many trees, how many nuts are we going to have to make in order to create seed orchards, in order to get blight-resistant populations out into the woods. So recognize that, here at the beginning, when we make seed, only half of them or less are actually going to be resistant. So let's talk a little bit more about backcross breeding. So there are thousands of trees across Massachusetts as part of the seed orchard program. When those were initially established, the thinking was that blight resistance was controlled by 3 major genes. It was this boulder model that we knew that the genetics of blight resistance was very simple and was inherited easily. Well, unfortunately, that's not correct. That was why the seed orchards were established in the way that they currently are. The worst case scenario is that if your trait of interest is following the stand model, where you have thousands of genes, each conferring very teeny tiny amounts of resistance or amounts of influence into your genetic trait of interest. Luckily, we're not dealing with that. We're dealing more with a river model, a river rock model, where there are more than 3 genes. We've got some that are conferring very large amounts of resistance and some that are conferring very small amounts of resistance. We still don't know what genes control resistance and where they are. We do hope we're doing a lot of work to try and figure that out. But based on the current information that we have from genomics and lots of study, we think we're kind of around here. So we might be able to find some high influence genes that control resistance. The way that we're doing that is by collecting leaves from all of the best hybrids in our program, thousands of them, and genotyping their full sequences. And so I don't know if Kendra can tell me how many trees have been genotyped in Massachusetts, Rhode Island. So 100 trees in Massachusetts, Rhode Island. I think across the foundation, we've done about 2,000, and we will definitely be doing more. And then we need to compare those genotypes with the phenotype. And the phenotype is what the trees look like, what they look like on the outside. So we say, is the main stem still alive? After we inoculate the tree, did it survive? That's the most important one. Is the blight sporulating? Is the blight really happy? We don't want it to be happy. Are the cankers sunken? Are the cankers big? How much of the stem is cankered, et cetera, et cetera. So we compare those metrics that we've been taking on thousands of trees, and we compare them to the underlying genotype. And by doing that, we should be able to hone in on the specific locations that are controlling resistance. At least that's our hope. This was me and Eric Evans doing this on some trees in Maine, and it's really hard to see. I'm sorry you guys can't see these slides really well. But this is a really small canker right here too. These are some Graves backcross trees in central Maine that look really fantastic. So what the original hypothesis with the backcross program was that we were going to find hundreds of trees that look like this. And again, unfortunately, we're only finding dozens. We're still finding them, but we're not finding them in as many orchards as we had hoped. So that's one element of breeding. We can look at the backcross, we can look at resistance in backcross trees. That's coming from Asian chestnuts. The other source of resistance that we can look at are from large surviving American chestnuts. And when I say large, I mean greater than 10 inches in diameter. And when I say surviving, I don't just mean alive, I mean surviving with blight infection. And so this tree right here, this is the Walbridge tree. This is up near Erie, Pennsylvania. And I always have to show off, that's Parker, she's my daughter. And so she's standing in this tree. Do you guys see how scruffy that bark is on the outside? So all these large surviving Americans, or what we call LSAs, all have this scruffy bark. They don't have that nice clean bark that you see on those really more, dare I say it, aesthetically pleasing Americans. Usually when you find those clean barked Americans, they're not resistant. When you see these scruffy bark or scraggly bark, these typically have resistance. That's not my daughter, that is Jay Brenneman. He's standing in the center of the Ort tree, which is another large surviving American in South Central PA. That's Tom Scielli, and he's standing at the base of the Thompson tree in Virginia. So there's a handful of these trees of wild type American chestnuts that have resistance that we've incorporated into our program that can enhance resistance of the Asiatic backcross hybrids that we're doing, and also from the transgenic trees that we're working with. And again, we want to pyramid all of these resistances together. We can deploy them individually, but we can also deploy them together to enhance the puzzle. Basically, the idea is these organisms, the chestnut blight organism or the Phytophthora cinnamomi organism or any other pathogen or pest that we come across, you want to give them as difficult a puzzle as possible to figure out so that they don't overcome the mechanism of resistance. And so that's the main reason why we follow this 3 Burr model, why we want to incorporate all of these different methods in our restoration population. I'm going to actually skip that. I'm going to talk a little bit more about best by best backcross and transgenic diversification. So how many of you guys have done controlled pollinations of chestnuts? All right, a fair number of you. How many of you have never done it but would like to? All right, fantastic, because we're going to need a lot of help doing these best by best crosses. So again, when the backcross program first started, those trees were all created by doing controlled pollination. You got to get up into the tree, you bag the female flowers, you come back 2 weeks later and you put the pollen that you want to get mom and dad together, and then you harvest those nuts afterwards. And so that's about 3 different trips to a tree that you got to take. It's very time intensive. It's very labor intensive. So the foundation did that a ton to make all the first backcross orchards that they did, and then was hoping that we would be able to do it with open pollination. Nature does this way better than we do, as you might expect, and I'll show you those numbers here in a minute. But those first 3 elements of what we want the chapters coming up to do, these new orchards of wild type Americans with Gcos, these best by best crosses, these transgenic crosses, these often almost entirely rely on controlled pollination. And so what we're going to do in the next couple of years is really focus on training new folks on how to do controlled pollination and then how to plant these into different types of orchards. Pollinations can be done by anybody, but really the first step you need to do is figure out how to get up into that tree. So a lot of it's done on ladders, although that does come with some risk. Getting bucket trucks or getting lifts, pull behind lifts is generally safer. We've had a great collaborations with Bartlett and local electric company co-ops. They've been fantastic in terms of donating their time to get to trees. But you can also, I know in the Pennsylvania chapter and the Pennsylvania, New Jersey chapter, we've been renting tow behind lifts, and that works great too. We rent them for about 2 weeks at a time to go out and do these controlled pollinations. And so, as I said, nature does this a ton better. So here, these orange bars are all the controlled pollinations that the foundation has done since it started work just before 1995. And you can see how low these are. So in its best year, in 2004, the foundation made just under 20,000 nuts with controlled pollination. In its best year, nature did over almost 170,000 nuts in a single year. So you can see the difference there. And that's a whole heck of a lot easier. And I'm sure any of you who have done the controlled pollinations, you realize that it's a lot easier to harvest after nature's done her thing than to do it yourself by hand. Looking at open pollination production, a question that I get a lot is, when do chestnuts start flowering? When are they going to produce nuts? And then what kind of production are we can we figure on? And so based on open pollination production at Meadowview, so Meadowview is in Southwestern Virginia, and that's the American Chestnut Foundation's main research orchards in Southwestern Virginia. We've got some really great data on harvest from the trees there. And you can see it fluctuates a lot. I think the main interest are this nut per tree metric. How many nuts are we getting per tree? So at age 5, very few trees were producing nuts, and we got a very low nut per tree harvest. Last year, we got almost 600 nuts per tree from the ones that we harvested. So you can see how that increases over time. We had a bumper crop in 2014 where, even though they were only 7 years old, we still got about 500 nuts per tree, and then that reduced over the years per tree. So the bumper crop was 2014. They got older, but the production per tree got lower just because that 14 was a bumper crop. And then last year, at about age 13, we got 600 nuts per tree. So we can start to look at the variability and start to calculate out what kind of production we'll be able to make into the future. But in general, the average age at first harvest is about 6 years old. Now, some trees will give you nuts earlier. So we've had trees produce nuts at 2 years old. We've had trees wait until they're 16. There are still trees out there that have not produced any nuts, but the great majority of them will start producing at age 6. Between 6 and 10 is generally when you can get production from chestnut trees. So how many bees can DACF control pollinate in a year? So this chart right here is showing how many nuts each chapter produced in the best year of production it had. And I do want to brag on the Massachusetts Rhode Island chapter, which has done really amazing work controlled pollination wise. It's, I think, the second, maybe third best production, annual production in the chapters. And that's not quite fair because that's following the Pennsylvania, New Jersey chapter, which has the help of Penn State and paid staff, and Meadowview, which also has paid staff at about 4,000 nuts. So as an individual chapter, Massachusetts, Rhode Island has had the best controlled pollination production of any other chapter. So you can see about, in its best year, over 3,000 nuts, about 1,000 bags, and so a controlled pollination efficiency is of over 3 nuts per bag, which is pretty good. Going back to the GCO concept, this germplasm conservation orchard concept, most of these pollinations were on wild type trees. So on wild type trees, you get about between 2 and 3 nuts per bag controlled pollination. Look down here at Tennessee and the Carolinas, they're getting it like 9 nuts per bag. And the reason for that is because most of the Tennessee and Carolinas production were on orchard trees. So this is another reason to have trees and orchard, and doing controlled pollinations in the wild is a lot of fun. Like, I like, who doesn't like careening over a hill at 90 feet in the air, you know, to do these controlled pollinations, but it's so much easier to do in orchards. So that's why we're really pushing this GCO concept for these best by best backcross crosses. It's going to be a lot easier to do those because they're in orchards than a lot of the early work we did out in the wild. We're going to get a lot more nuts than we used to, and it's going to be a lot easier. So once we get these orchards established, get production going, we're already doing reintroduction. I know that there are some reintroduction sites here in Massachusetts, Rhode Island. This is in northeastern Pennsylvania. This is a hunt club called the Blooming Grove Hunt Club. This is a site that was established in 2017. This is a wild type American chestnut stem that's planted, that's in the middle of this planting. This is a site that was established in 2015. I took these pictures this spring, so you can see some really phenomenal growth on these trees that were established here. These are all advanced backcross B3, F3s, if you guys are familiar with that terminology. That's what's planted here. We've got maybe 50 of these reintroduction silvicultural type trials planted across the eastern U.S. that are giving us really good data about how to put these trees back in the forest and what kind of resistance we're getting out of that material. There were an estimated 180 million acres in the native range of American chestnut. This is Little's range. This red mark is all Little's native range. If you look at, don't look at climate, if you look strictly at various soil aspects and suitability of an American chestnut range in the eastern U.S., so east of the Mississippi, we're looking at about 100, I'm going to round up, we're looking at about 100 million acres of suitable habitat for American chestnut across the eastern U.S. So the native range is about 180 million acres. We're obviously not going to plant all that. Let's say 100 million acres of that is suitable. Look up here in northern Maine, where there currently are no American chestnuts, but that land is highly suitable and will be as climate change continues. So how do we figure out how to get these trees not only planted, but then self-sustaining and self-populating? There are a few of these sites where we found American chestnut reproducing itself. West Salem, Wisconsin is the most famous. It was profiled in the late 80s in a National Geographic article in 1989 talking about, there was a farmer who basically stuck 9 seeds in his pocket, went out west and planted them on his farm in western Wisconsin, and those 9 seeds grew into 30 acres of chestnut. So in terms of figuring out how chestnut spreads, this is really the best place to do that. That site has since I think starting in 2015, I think all the original trees were dead, and now most of the second, and third, and fourth generations of trees have succumbed to blight subsequently. So that site is no longer a great place to study. The Kendra Pose, if you guys remember that, that was taken here in Berlin, Vermont. Those trees have now succumbed to blight. Atkinson, Maine was a fabulous place to study. It was about 15 acres of American chestnuts. I was there a couple weeks ago, and unfortunately that site has now completely succumbed to blight. This Orleans site has natural regeneration. Rufin, are those still doing okay, or have they succumbed? They're doing pretty good. So we've still got some trees there that are naturally regenerating. And then Roselawn, Indiana, that site got blight a couple of years ago, but I'm pretty sure, I'm gonna knock on wood, the Indiana state champion is there and is still producing nuts. So we're sort of racing against the clock to get some of this information about how chestnuts spread. Unfortunately, it wasn't documented in the early 1900s because that was really when forestry was coming into its own. And when then the blight came and we weren't able to get a lot of data on regeneration potential of the species at that time. So we're kind of racing against the clock to get West Salem documented, Atkinson documented. We've got data since 2011 on that site, and should be publishing a couple papers on that. And then the Roselawn, Indiana data, we should be getting some great information there. Here's some old pictures of West Salem. This is from the early thousands. Here's what the old trees look like, about 100 years old. And then these are all American chestnuts that rose up from those very few trees. Based on the West Salem, Wisconsin site, now take this with a grain of salt. Based on modeling regeneration of the West Salem site, Steve Rogstadt and Pelican, these were researchers at the University of Cincinnati. They were using a model called New Garden to try and figure out what does a restoration or a reintroduction population look like. In order to have the most allelic diversity of what you plant, how many trees do you need to put into a plant? And so what they found was, in order to get the greatest population growth for allelic diversity in a breeding population, you need about 169 trees. I round that down to 150, because in order to get those 169 trees, based on the data at West Salem, they found you needed to have about 300 trees in the beginning. And what I find really interesting about that, that's what we find in our orchards. So the Chestnut Foundation has been planting trees since the late 80s, mid 90s, and we find that our orchards generally have about 50% survival long term. And interestingly enough, that's what we found at West Salem, is when you establish 300 trees, you typically get about 150 of them breeding long term. And that's how you get the greatest allelic diversity. Generally, I'm going to recommend, if you want to do a reintroduction type trial, I'm going to recommend you plant 300 trees on about an acre, which is generally pretty good forestry spacing. Some people are planting 500 trees per acre, some people are planting less. That doesn't mean that you can't plant 3 trees in a gap opening or 2 trees in your backyard. That's fantastic. I'm going to encourage that people plant any and all kinds of types of plantings, because the job we have ahead of us is just enormous. But in order to follow some sort of standardized design for increasing allelic diversity across time, I'm going to recommend planting 300 trees per acre. I'm going to recommend that's a monoculture of chestnut, and that's going to give us about 150 trees breeding to one another. That's going to be a reintroduction cell. All right, so keep that in mind. That's not going to preclude other models. Other types of planting are absolutely going to be valid and work. But if we use this as a starting place, keep that in mind. 300 trees per acre, giving us 150 long term. How do we get that? How do we get that many trees into production? So we've got containerized methods. This is the Valonia State Nursery in southern Indiana. It's just a gorgeous place. They get 7-foot seedlings in 1 year. They are just phenomenal. This is the Penn Nursery orchard, or Penn Nursery is in central Pennsylvania. It's the DCNR nursery in central PA. Seedling production is really difficult. There are 1.2 billion seedlings planted on 2.5 million acres in the eastern U.S. every year. 82% of those 1.2 billion seedlings are planted in the south. 75% of that is their rootstock. Anyone just yell out what you guys, and if you see my presentation already, you're not allowed to yell out. But if you haven't, what do you think most of those 1.2 billion seedlings are? Any guesses? What makes the most money? Yes. Softwoods, but do you know what kind specifically? Loblolly pine. That's about 1 billion of those seedlings are loblolly pine. And that's just because they make so much money and they're planted all over the place. Hardwood seedlings are in demand, but they cost a lot more to grow, they cost a lot more to install, and they take a lot more effort to get growing. And American chestnut is a hardwood tree. Unfortunately, we're not dealing with something. I think if we were dealing with something like poplar or pine, we might be done already. So 5% of that production was hardwood trees. So instead of 1.2 billion seedlings, about 1 billion of that, which is loblolly pine, we're looking at 22 million trees every year annually. That's hardwood production on about 43,000 acres. That's the annual maximum production in the eastern U.S. for hardwood planting. 67% of the hardwood production was oaks. So not only do we have the blight resistance that's really difficult to get to, but we think we've got a really good methodology of that right now. Now you're facing, how are we going to get just thousands and thousands, millions and millions of seedlings created and planted out on the landscape? Let's say we can get to 2 million seedlings per year, which right now, so based on that last chart of open pollination, our maximum production per year was 170,000 seeds. So we've got our work ahead of us to even get to 2 million seedlings per year. But let's say we can get to that. Once we get to that 2 million seedlings per year mark, let's say we can cross at 300 trees per acre. Let's say we can get 4,000 acres per year. That would take us 250 years to cover 1 million acres. So that doesn't mean that we can't plant more across more acreage, but even to get to that 2 million seedlings per year milestone, it's going to take us a couple decades, by my estimation, to get there. The reason I use that 1 million acres is because, remember before I said about 100 million acres of the eastern U.S. is suitable habitat? Well, 1 million of that is about 1%. So if we say, how much are we actually going to plant and how much are we going to let nature take its course? So let's establish a goal that we want to actively plant about 1 million acres, which is about 1% of suitable habitat, and we'll let nature take care of the rest. That's still a huge amount, because even if eventually we're going to create more than 2 million seedlings per year, but it's gonna be an uphill battle to create even that, and it'll take us a few decades to cover that 1 million acres. Some chestnut habitat is clearly not going to be suitable for planting. So this is Greg Turner. He works for the Pennsylvania Game Commission, and he's working with Allegheny wood rat, which is an endangered rodent species that thrives on American chestnuts. That's its preferred food source, and it's endangered because its preferred food source has been virtually eliminated. This is where it loves to live, just in rock piles. And you can find, this is the tree, we call this the rock dump tree, for reasons that you can guess, because it grows in the middle of rock. I don't expect that we're going to be planting in stuff that looks like that, although I have planted and stuff like that. I don't expect that that's going to be where we're going to plant these reintroduction trials of 300 trees. I would rather let the wood rat do that. So we kind of plant near these rock dumpy areas, and that the wood rats and the squirrels and the blue jays repopulate highly suitable habitat, but where we don't really want to be working in directly. So just a better picture of trees growing in rocks. And the way we get there is through these regional programs. So just training people how to do pollination, so we can establish seed orchards, so that we can get to the hundreds of thousands and millions of seedlings that get produced. These are all the orchards, the breeding orchards that we have. So the Chestnut Foundation has a history of collecting nuts, of planting them, of having production. We can get there, but we have to have all hands on deck, producing seeds, planting seed orchards, and getting them producing material to go out. And then one day we can all be doing the Kendra pose by some of these big trees. Again, this is the Berlin tree, this is the tree in Atkinson. So this would be more or less a big parent tree, about 100 years old. This is a tree that's more like 40 years old size. These trees produce tens of thousands, hundreds of thousands of nuts. And we have to start now really to get to that point in the future. We've got best management planting practices. We've got guidelines to put these trees in the ground. This is a reintroduction planting that we've got about an hour east of Pittsburgh up on Chestnut Ridge. What are the best ways to reintroduce this? This is really going to be case by case specific. Even though we have BMPs, do you want to plant seeds or seedlings? Do you want to plant bare root or containerized? Do you want to plant in a forest or a field? How much do you control the vegetation? Do you use herbicide or not? All of these things are going to be case by case. We have recommendations that we can give you. That's what Kendra and I get paid for a lot of the times, to make these recommendations. But really it's going to be up to an individual land manager and what they can implement long term. We've learned a lot based on wild Americans. So these are 10-year-old American chestnuts, no resistance at all, where we did seed versus seedling, containerized versus bare root, herbicide versus no herbicide, tree shelter versus no tree shelter. We got some really great data on these sites. What's really interesting about these trees is they never flowered. They got to be 15 years old before the blight put them out, but because there was so much shade from competing trees, they never flowered. So one of the biggest things that we've been learning from these types of plantings is, yes, you can get establishment, but if you want production from the trees that you plant in the forest, you want it to be fully open to get these trees reestablishing themselves in the woods. So you can plant in gaps, you can plant in shelter wood. The trees will do great, but they're not going to start producing until much, much older. You definitely want to you want to control the competing vegetation, but in some cases chestnut can do it. This is all of this is stump sprouted black birch. But the American chestnuts, you can't see that. There's the pole for the American chestnut. That's Tom. He's measuring American chestnuts. The American chestnut can compete against some of this other stuff, but they'll do better if you control it. So generally, we recommend controlling competing vegetation for 5 years post-establishment. And this is a glyphosate. This is, some people use cardboard. Some people are following organic methods. That's fine. But generally we recommend 3 to 5 years of vegetation control post-establishment in order to get these things going. This is the one seedling that survived. This was a direct seeding experiment. We took 40,000 nuts and broadcast them across the field. There was no vegetation control done with that. There was no rodent control. We had one tree survive. When we used vegetation control, we had 10% survival of those 40,000 nuts. So it really goes to show how much this vegetation control is going to be so imperative for long-term installation and management. We've got lots of other plantings like that. This is the Hoosier National Forest in southern Indiana that we're getting data from. This is the Monongahela National Forest where we're getting data from. We've got a lot of different documentation. If you guys are interested or if you want these, we've got the Task Force Protocol. We've got the SFI, Best Management Practices. We've got a bunch of fact sheets that can help people get started in learning how to plant trees. So looking beyond seed orchards, looking at production, when can we get to that 2 million seedling per acre time point? So we've got Darling 58 that should be deregulated in the next couple of years. We've got our best backcross crosses that we're looking to plant and put into production. We've got about 100 acres so far that we've put into reintroduction plantings with our backcross trees. So that's where we are right about now. With the material that we currently have, I'd say we could plant about 500 acres by 2030. That's when we should have the wound-inducible promoter. If you guys are on the TACF listserv, you guys probably saw this paper that came out from BSF on the wound-inducible promoter, the Darwin that they're calling. That paper just came out, and we're going to stack resistance. We're going to backcross trees with Phytophthora resistance, with OXO. We're going to start stacking all this together. That should come into production in 2030. We should reach about 1 million seedling production by 2040, which means we should be able to cover about 5,000 acres in the next 20 years. Maybe we'll have gene editing by then. Hopefully we can figure out what genes are controlling resistance in the next 5-10 years, and we can implement things like CRISPR or whatever's on the horizon in the next 20 years. And we'll have those stack resistance crosses going out. By 2050, maybe we'll have been able to plant 20,000 acres, and in the next 40 years, I think we will have been able to cover at least 50,000 acres, and we can reach that 2 million seedling production goal in the next 40 years. But that all depends on us making those crosses now, establishing those seed orchards now. And I think the other point that I want to make is everybody's really excited about Darling 58. A lot of people are making crosses of backcross trees or have already planted them in the woods. But, and they're going to play a really important part in terms of restoration. But I think we always need to keep our eye on that there's always going to be better stuff coming out. That doesn't mean that the stuff isn't good. It just means that we're going to have improvement. We're going to have incremental improvement as time goes on. We're going to need to establish seed orchards of this material or cross the material in our seed orchards with this improved material as time goes on. And because restoration is going to take decades, it may not take as long as 250 years, but it's going to take 100. You know, it's going to take a long time to see an ecological impact of what we plant. And for better or worse, and I think for better, that means that we're going to be able to implement all of these new technologies over time and improve our restoration populations over time as well. So with that, I'm going to share, I love this picture. This is an American chestnut looking over the Southern Appalachians outside of Asheville, North Carolina. Ben Jarrett took this picture, and I think it's really fantastic. And I think that's really what we're aiming for, is to have American chestnuts once again providing these ecological services in the landscape. So thank you.
If you have a question, it would be nice if you came up towards this little microphone, and then people on Zoom will hear it. So if you want to queue up here for questions instead of staying at your seats, that we don't have to pass this around and stuff.
I would like to know just where we are on regulation, and could we have a picture of that, please? Thank you.
Sure. So the regulatory process for Darling 58, for those of you who don't know, it was started in January of 2020. The USDA got the petition from SUNY ESF to deregulate Darling 58. That was then put out for public comment in the fall of 2020. In the fall of 20, and the public comment was generally positive. Over 60% of commenters stated they were in favor of deregulation. And most of those were highly substantive, provided the USDA with good information of why this material is safe. In the end of 2021, so August of 2021, the USDA submitted an NOI, a notice of intent. And the notice of intent basically says, hey, everybody, we got this petition. We're going to develop an environmental impact statement, an EIS. We're going to do that in the next 2 years. What do you guys think about that? And so there was a lot less interest in NOI, but what that told us, the ACF and ESF is the USDA is planning on giving their environmental impact statement within the next 2 years. That's not the decision. The decision will be made shortly after that. So we're kind of running on, some people think it's going to be next year. Some people are very optimistic that it's going to be next year. I think it's probably going to be another 2 years. I think we're not going to see a decision until the end of 2024, but we don't know. That's the USDA. The EPA has their petition that they're evaluating. They have 18 months from the time that petition is submitted. So the EPA should make their decision by the end of 2022. So I think we're going to not see a decision until the end of 2023. It's probably the earliest that we'll see a decision. Some people think it's going to be next year. The EPA should make their decision at the end of next year. It's going to be in the next 5 years at a minimum. In the meantime, anything that we work with the transgenic material has to be under permit, and that's what we're doing. And then TACF is working really closely with ESF to develop distribution pools of seedlings that can go into seed orchard production, that can go out to distribute to long-term members and supporters right upon deregulation. Is our hope is that we'll have material to give to supporters the second that we get deregulation notice.
Hey, Paul Marino. So many questions for you, but I'll just choose one. In these populations, like not Wisconsin, because that's a totally interesting, different topic. It's like basically like an invasive plant there, right? Just like say black locust in New Hampshire, even though it's native to a nearby state. But it was released from its enemies, right? But in with the populations that have been, in the Northeast in the native range, with your data that we're gonna, will your data include Cryphonectria there? And as you're making this, as you're on this trajectory, hopefully things in nature continue to exploit Cryphonectria get better and better at exploiting and parasitizing Cryphonectria. And are you seeing, are your data showing any of that? Are there any new examples of naturally occurring hypovirulence or in any of these stands that are doing a little better or a little more reproductive than in other places? Have you noticed that Cryphonectria there is maybe a little weaker or has more enemies?
That's a great question. We're not really seeing that. We do see hypervirulence making an impact, and that's why it is going to be one of our strategies, I think. So the Chestnut Foundation is going to be deploying a super donor experiment next year at some select locations. So the super donor is a hypervirulent organism that's designed to spread the virus to more strains than it can naturally. So we're going to experiment with that a little bit more to see if we can't deploy that in greater number. There's still the cruddy bark phenomenon that we do not understand. We don't. If you guys aren't watching the Chestnut Chat series, I hope that you guys will. But all of our Chestnut Chats are recorded, and there was a really incredible one done this past January that was all about cruddy bark. So there's a natural phenomenon. It's not hypervirulent. It's something else. The trees that have cruddy bark look a lot like those large surviving Americans I showed you. So you've got resistance that's certainly a component of very few American chestnuts. But then there's something else that's controlling blight. We don't see it spreading, but it's certainly cropping up in more places. And is that because we're noticing it more or what? We don't know, and we still don't know what it is. But I think we are going to see more and more of these biocontrols that we can be able to deploy and implement as part of our long-term strategy. So the question for those of you on Zoom, the question is, are the trees at West Salem being killed by the blight? They are. The stands toast. I mean, all the big trees are dead, and blight has pretty much taken over on the stands. They're dead. Unfortunately.
Come over here to ask your questions, you can. Then people can hear it. We can line up along here.
Okay, I had not previously heard of the cruddy bark. I'm just wondering, considering that it might not be directly related to the resistance, but it might be from a gene, a cruddy bark gene that is simply close to, located close to a gene for resistance.
It could be. I mean, we have no idea. And so we've got people who are studying the cruddy bark from the standpoint of shotgun genomics. We're just throwing that stuff and looking at what kind of organisms are in there. And we have not yet evaluated any sort of underlying resistance of the trees that are exhibiting cruddy bark. Currently, the cruddy bark appears to be a non-resistant. It looks like it's some sort of other organism that's antagonistic to Cryphonecria versus resistant to the American chestnut. But that doesn't preclude that it is.
Hi, at the New England base camp, we have at least 50 chestnut sites, and quite a few of the trees are 20 feet or taller, but there's competing vegetation around. Is it none of them have yet produced catkins or nuts or anything like that? So I'm wondering, should we try to clear away some of the competing vegetation?
If you want nut production, then yes. And so usually it takes about a year. So if you cleared around it now, you wouldn't see nut production this year. You would see it the year following or even 2 years following. But yeah, so, in order to produce cactus, the flowers, chestnuts need full sunlight in order to do that.
Okay, thank you.
The disadvantage, I will say there's a disadvantage, because once you release chestnuts, if they don't have any resistance, they grow very, very fast, and they tend to get infected much more quickly than the slower growing trees. So that's the trade-off. We've done that to a lot of wild American chestnuts where we've released them and gotten up production, knowing full well that they're gonna die a lot sooner. Generally, the philosophy is we would rather rescue that germplasm and have it used for other things than just keeping the trees in the vegetative state. But that's the disadvantage.
Yeah, because most of them have died off anyhow.
Yeah, so if you want to get enough production, release them, and you'll get about a good 5 to 8 years of production before they become.
Okay, thank you.
Yep.
Masks and glasses don't go well together. I have numerous questions, but I need to pick one or two, I think. One of them is, you said it'll be about 100 years before we have some good restoration. I was planning on living to be 100 years old, but I'm already headed close to that. Does that mean I'm going to have to try and hang on for another 100?
I think if you can, but no, I think what it means is, we're gonna have we have restoration populations right now. We have populations that are in the forest that have resistance, and we're gonna have more of them. What I'm trying to say is an ecologically impactful landscape-wide restoration is what I'm talking about. That doesn't mean that we're not gonna have restoration populations in place. We will, and I think in order to start, so you've got this small scale look at people putting in these plantations or grows this year, next year, 10 years from now, those all add up over time. And we say, when are we gonna see a landscape-wide, when are those populations gonna start breeding together and having self-sustaining populations? And that's what I'm looking at, 100 or more years into the future. But initially, you've got to start with these smaller reintroduction populations that are making the impact locally.
Okay, good. I'm going to work on living to be in good health, if that be also.
Yes.
So I'm working on 100. I think I'll do that probably, but
Beyond that.
I'm hoping I see something that looks good by then. The cruddy bark thing, I've seen that on some Americans that I have interspersed with a orchard of F1s that I have. It looks like kind of a webby sort of thing in the bark. I thought I had a couple of trees that were goners. They died back, but now they sprouted up again. They're taller, and a couple of them put out lots of pollen. So I've got male sterile F1s, and they pollinated the male sterile F1s, and I got great yields on those trees. So I find that interesting. The Salem thing, I thought I had heard, as Mike was mentioning, that there was the hypovirus was in that stand, but it sounds like that really is not proven out.
So the hypervirulence that was deployed at West Salem can keep individual trees alive for an extended amount of time, but not forever.
Okay.
And so the data that they found from the West Salem deployment saw that it can go from canker to canker within a tree, but cannot spread from tree to tree. And then eventually you get a canker from a strain that hypervirulence can't control. So the super donor strain of hypervirulence was not deployed at West Salem. The super donor has been deployed in Western Maryland. And so we need to look at more data of a strain of the fungus that can infect every strain that's out there. That was the downside of what was done at West Salem. And so hypervirulence can work, it can extend the lives of trees, but you need a strain that can infect all strains of plant. And I'm seeing a lot of chat here too. Is there anything in there? No, that's all. There's no question.
I have stand in Wilbraham, Massachusetts that I mentioned. Near that stand is a high school, and they have some land where they have cross-country trails. The tornado went through in 2011, knocked down all the competition, not all of the competition, but a fair bit of it. And the trees that remain look like palm trees, oaks that got, now they're starting, some of them are filling in below. Some pines that survived. But it opened it up enough that I found about 50 Americans in that area. And they were producing pollen. They got up to about 30 feet tall when they were doing this. The growth rate was about 1/2 inch a year. Those trees, I think, are in fair shape. They do have blight, but it looks like an interesting bunch of plants, American trees. And Mike has, and David have collected a number of those nuts, have them in our conservation orchard, the one that they're working on.
Yeah.
Now, there is one tree that got up to about 10 inches in diameter. It's got a lot of blight in it. How much longer it's going to last, I don't know. But the stand seems to have bigger trees than I see a lot of other places. There are reports of some that were in a brimfield orchard. And my understanding is they grew fast and died back fast. So the stand that I'm looking at is of some interest, I think.
If I can just address that. So disease, are you guys familiar with the disease triangle? So there's to get disease, like chestnut blight, you need 3 things. You need the host, you need the pathogen, but you need a conducive environment. And so I think a lot of the differences that people see in things of chestnuts living longer or not as long, a lot of that is the environmental component. So we can't discount that. And I think as we talk about restoration and introduction, we need to be very cognizant that some of these restoration populations are not going to do well because they're in a bad environment. And when we go to plants, we need to give them the most optimum location as possible, because even if they have resistance, even if it's transgenic, or even if it's backross, or even if it's all these combined, if you don't have the right environment, or if they're stressed because they're in a poor environment, they're not going to do well. So I think we always need to do, and so I think a lot of what you might be seeing and what I've seen in wild stands that get released is environmental, of when they get infected faster than not.
One last thing I think I'll mention, we might be able to talk after or during lunch,
Sure.
something like that. The one thing that I have noticed with the trees that I've had, as long as the bark on the chestnut tree is smooth, the trees are fine, except that in every lenticule, you'll see a little orange dot. On occasion, you see 2, and they're all sitting there. And when the bark begins to crack, it follows through those lenticules. And by golly, that light fungus gets into the bark area of the cambium layer. And I have been concerned about the test that we do, in that we get right through and into the cambium layer. The question, I think, is, could it be that cambium layer is also important in terms of production of bark? And if it produces bark fast enough, it will tend to protect itself from the crack getting all the way into the cambium layer.
And I think that's a really important point. And one of the reasons why we have these long-term tests established in the field, to make those comparisons from artificial inoculation to natural inoculation. So based on what we've seen, so a lot of people are concerned about it. It's not just, so the tests that we do with artificial inoculation, we bring the hammer down these trees. That is absolutely correct. I think that we are, and some people are concerned that we are hijacking some natural defenses that we can't see through that artificial inoculation. That could be through what we're seeing in our natural populations that have been out since 2008. we're not seeing that. We're not seeing a loss of any of these types of mechanisms, and those mechanisms are incredibly complicated. It's something that we are cognizant of and that we're watching very carefully. But I think that it number one, shouldn't stop us from doing artificial inoculations, but number two, I want us to be sure that we're continuing to be aware of that mechanism and continue to make those comparisons of natural inoculation versus artificial inoculation and see if those two correlate long term.
Well, in my F1 stand, I have not done artificial inoculation. I've just let trees grow. And that's it. So I do have a stand of F1s where that's going to be the case. All right. I'd like to talk with you more.
Okay. Thanks, everybody. I appreciate it. I think we have one.
Yep, let's done.
For the folks on Zoom, we are,
Yes, so we're in the lunch period now, and you can slowly make your way over to the buffet table. Keep your distance, enjoy.
okay,
Back in about an hour.
and I'm going to show you the room real briefly for the folks on Zoom. If you want to, let's see, yeah, put the speaker. So I'll have me be the speaker, and I'm going to show you the room here. Oops, if I can. So it's not really maximizing my face, but maybe you can still see a little bit. So here's one of the usual suspects here saying hello to you all. Okay, so let's see. Hey Buzz, did you see that? Buzz was interested. Anybody else? Okay, great. Thanks, Maria. Hello. I hope everybody there is doing well. Great. Hi, Buzz. Hi, Maria. Hi, Bill. David, Ellen, Thomas. Good to see you here. And Eric and Jamie. Okay. So hope you folks are all doing well. So we have a slideshow that I'm going to attempt to show during lunch. And so this will involve me sharing my screen and that kind of zoomy thing. So I'm going to try to do that. So I'm going to try to do my slideshow, and then I would share my screen.
Okay.
And we'll see if that works.
USB or
I have
C.
Okay.
Old, I
so-
think.
Okay,
Old
so we
fashioned.
don't need to do
Okay.
that.
And
Yeah.
we are attempting
What do
to
you want?
monitor chat, by the way, if
What's gonna
anybody's
be-
watching.
What's the
that
easiest for me
Wants
to?
to ask questions.
Yeah.
I could just plug in here with my computer too.
You could do that too if you want. But then you have to be part of the Zoom for people to see it.
I have to be in Zoom. Okay.
Yeah, so it's,
Um,
I can do it from this one here. Just,
So if I'm understand that, where do I, if I'm standing at the
you
podium,
can stand up there and just give me a clue and I'll just flick it if you want. Or we just should have a,
I could do
our
that. I could
thing doesn't
pause
seem to
record
work.
it. Yeah.
our,
You also
let me just find it here.
I mean, it's really up to you, then you'll have peace, but yeah. Like giving Brad or talk.
Yeah, I could find it here.
well, I'm trying to figure out the logistics. If they're gonna be standing at the podium.
Oh.
the presentation goes here, that's it.